Good afternoon, students. Here you have to enter, like when you're entering in the Zoom app, please give your name. Okay. I don't know this Oppo 5F youth. Who is it? Please tell to put the name so that I can enter them. Okay. Okay. Good afternoon, students. I hope you can see the presentation. Yes?
Yes, ma'am.
Okay, fine. Last class we have studied this thing, Volvox and Udgonium. So I have completed three species, that is Nostoc, Volvox and Udgonium. Today we will see about Sargassum, hopefully Batrachospermum also. Now we'll start about Sargassum, the fourth species. Now this Sargassum belongs to kingdom Chromismeta, phylum Orchophyta, class Phaeophyceae, order Fucales, family Sargaceae, and genus Sargassum. So this picture here, it represents Sargassum. So this is how the Sargassum looks. Okay, now this Sargassum is commonly called as gulf weed. Okay, so it is a brown alga. It is included in the class Phaeophyceae and in the family Sargaceae. So this Sargassum, it was first reported by Columbus. Okay, the scientist Columbus in 1492. So he derived this Sargassum from Pacific Ocean. So he got this Sargassum from Pacific Ocean and he named Sargassum from Sargasso Sea. That is in Pacific Ocean there is a sea, Sargasso Sea. From Sargasso Sea he gave a name called Sargassum. Okay, so this is most abundant brown seaweed in India, that is Sargassum. So if there anyone asks you which is a brown seaweed which is available in India, then it is Sargassum. Okay, so Sargassum is a brown algae. It is included in the class Phaeophyceae and in the family Sargaceae. It was first reported by Columbus in 1492 from Sargasso Sea, a part of Pacific Ocean. Okay, this Sargasso Sea, it is a part of Pacific Ocean. So this is the abundant brown seaweed in India, that is sargassum. Okay. Next, its introduction, like its distribution or its occurrence. Sargassum, it is a marine algae which is popularly called as gulf weed, okay, or gulf weed. The sargassum plant is macroscopic, more or less bushy in habitat. We have seen the diagram here. So this is more or less bushy in habitat, and this is macroscopic. Means it can be seen through your naked eyes. It is not micro, but it is macro. Most of the species occur in tropical or temperate zones attached along the rocky shores. Some species occur along the Indian coast, that is east and west, and some of Australia Sea and also seen in Japan. So, in all, sargassum is a common inhabitant of tropical seas and oceans. Like you can see, sargassum is normally found or it is occurs in tropical seas and oceans. Okay. This includes about 250 species. Sargassum alone has about 250 species. So out of some of them which are common in India, I have listed, that is Sargassum tenerimum, S. chirophyllum, duplicans, paleophyllum, isilifolium, and mycosystem. So these are some Indian sargassum which we find in India. Like these are species of Indian species. Then about its structure or what it is, how it is there, how the structure of sargassum is there, or how it represents. So sargassum, as we seen, it is a brown algae. It is commonly called as gulf weed or it is seaweed. Then it is included in the class Phaeophyceae. It is a marine algae. Then it is attached algae. Okay, it is an attached algae. It grows up to 30 centimeters long or more. Okay, so it is a diploid sporophyte. That is why it looks like an angiospermic plant. Okay, that is why it looks like an angiospermic plant. Then its main axis is approximately 30 centimeter long or more. It bears richly branched primary lateral. The primary lateral has unlimited growth, so it is called as long shoot. So there are shoots. Shoot means stem, like a shoot of limited growth and shoot of unlimited growth. So the unlimited growth is called as a long shoot. So that the main axis has two types of branches, that is primarily called as primary laterals and secondary laterals. So the primary laterals are of unlimited growth and secondary laterals are of limited growth, and they look like leaves. Okay, and the branches are monopodial in arrangement, and the leaves have entire margin. You can see, just see the diagram, I'll explain it so that it becomes easy than seeing a diagram and hearing rather than just reading the points. Okay, so this is the diagram, you can just see here and we'll keep on explaining. So there are two types of axis, that is primary laterals and secondary laterals. The primary laterals form the main axis, and the primary laterals, they are of unlimited growth, and secondary laterals have limited growth, and they look like leaves. The branches are monopodial in arrangement. The leaves have entire margins. You can see the leaves here are entire like coconut. Okay, it are here the leaves have the entire margin. They have no midrib. So midrib is not there. Each leaf possesses a few pores on the upper surface, and these pores are called cryptostomata. Please write it down. Cryptostomata. That is CRYPTOSTOMATA. Tomata, that is cryptostomata. So what are cryptostomata? Cryptostomata are found on the upper surface of the leaves. These are the pores and these are flask shaped cavities. Okay, these are flask shaped cavities called cryptoblasts, and they are called air bladders also. Stomata, so but obviously you can just relate it to that. They are called air bladders. Now the air bladders are stalked, okay, and they are called as modified leaves. And now these stomata or the leaf, they give a buoyancy to the plants because they are also found in the sea. So these give buoyancy to the plants. Who gives buoyancy to the plants? That is cryptoblasts give buoyancy to the plants. Okay. Some lateral branches here, they repeatedly branched and are called fertile branched. So some are repeatedly branched, so they are called as fertile branches, and these fertile branches here are called as conceptacles or conceptacles. This is very important here to note it down. That is called as conceptacles because from this conceptacles only you contain, like the origin of sex organs takes place in conceptacles, that is a fertile branch. So this is about the thallus. We will just read the points here so that you come to know. From the first, sargassum is a diploid sporophyte. Then it has main axis of approximately 30 centimeter long or more. It bears richly branched primary lateral. Okay, that is a stem. Two types of stem I told you. This primary lateral has unlimited growth, and it is called as long shoot. Okay, the long shoot bears numerous secondary laterals, that is secondary stem branching. So they are called as limited growth. Of the latter, the basal one is usually most conspicuous. It is shaped like a leaf. Okay. Now, the basal member or the members of auxiliary branch system, they are swollen and where was I? Into a stalked and rounded structures called air bladders. I told you air bladders also called as cryptostomata. The air bladders contained the small gases at the atmosphere and helps to keep floating upon the surface of the water. So that is why I called it as a buoyancy. So these air bladders or cryptostomata, they give buoyancy to the plants so that they keep floating on the upper surface of the water. The subsequent members of the auxiliary branch system, it may be cylindrical or flattened. They are called as receptacles. So these receptacles, they are flattened and they formed in the nodes or internodes, and they form fertile branches, and these receptacles are, they have flask shaped structures which are called as conceptacles. Now these conceptacles, they have the origin of the sex organs which help in the sexual reproduction. So in sargassum, you have to remember cryptostomata and conceptacles. These are for 2 marks. Please write it down. For 2 marks, cryptoblasts or cryoblasts or cryptostomata or conceptacles. Okay, these are cryptostomata and cryptoblasts are the same. Conceptacles are different. Conceptacles are flask shaped structure which arise from the fertile receptacles. Okay, so here you can see the diagram. Now see the structure of sargassum. AB indicates air bladder. Here this is an air bladder. You can see my cursor. This is an AB indicates air bladder. Then RC, it represents receptacle. See here. This is RC. This is receptacle. Then AX is the auxiliary branch. Now see this. AX, it is an auxiliary branch. Then MD, this is a midrib. I told you leaf is entire with one single midrib. Then I, this is bearing lateral. Okay, this is lateral branch. Now here the structure of Sargassum longifolium. This is the main axis. Then this is a air bladder which is terminated by a leaf. Then this is lateral or a leaf. Then this is receptacular branch, and these are fertile receptacles which has conceptacles. Now this is one more species of Sargassum. It is called as nervi. This AB, this is called as air bladder. This see here, air bladder. This ABR is auxiliary branch. Then LB, this is BL, this is basal lateral leaf. Then I, this is lateral branch. And this is a main axis, MA, this is a main axis. And this is hold fast, the basal cell which holds the substratum. This is called as hold fast. So these are the three examples of Sargassum. Now, structure of thallus. Now this is the structure of thallus. Like how does the thallus looks? Okay. Now see the diagram. I will just go on explaining you. Now here it has a single layered epidermis, and it consists of loosely arranged thin walled cells, and these epidermal cells, they contain chromatophores. Okay. Now the transfer section, it shows outer epidermis, inner medulla, and middle cortex, also called as medulla. Okay. The epidermis, it consists of compactly arranged parenchymatous cells. Okay. The epidermal cells contain chromatophores and reserve food. You can see this pellucid layer which is below the cuticle. Okay. They are then the meristematic nature, so that epidermis is also known as meristoderm. Okay. The epidermis is also called as meristoderm. The medulla consists of thick walled cells. Okay. It serves as a conducting tissue. The cortex is present in between the epidermis and the medulla. So this is the cortex. It is present in between epidermis and the medulla. It is several layered in thickness, and it is made up of thin walled parenchymatous cells. So here you can see the cortex. It is made up of thin walled parenchymatous cells. The cells of the outermost layer of the cortex contain chromatophores, and they help in photosynthesis. The cortex stores reserve food materials. So this is a simple diagram of thallus. You can just see the points here. It is divided into three layers. That is upper epidermis, which is also called as meristoderm. Then you have cortex. Then you have medulla. Now meristoderm, it is a peripheral layer of columnar cells without any air space between them. It contains three layers of cell: pellucid layer, hypodermal layer, and cuticle layer. Then next comes cortex. Within the hypodermal layer, there is a cortex. It consists of thick-walled polygonal cells. So it is in between. Then you have innermost layer, also called as medulla. The central zone, it is called as medulla. Okay. The cells of this region, this is loosely arranged. They are narrow and elongated. The outer cells of the medulla have thick walls, and whereas the inner walls have thin walls. Okay. So just write it down, the structure of thallus. I'll give you a minute. It is divided into three. That is mesodermal, that is also called upper epidermal layer is also called as meristoderm. Then inner cortex. Then in between you have medulla. Then next, how is the reproduction? Now, the reproduction in sargassum, it takes place by two methods, that is vegetative method and sexual method. Now, how does vegetative method takes place? Now, this vegetative method, it takes place by fragmentation. Okay. Now, how fragmentation takes place? Now, the vegetative thallus, it breaks accidentally into new fragments. We have seen this fragmentation many a times. So I think there is no need of huge explanation here. So what happens in fragmentation? The vegetative thallus, it breaks accidentally into new fragments. Then these fragments grow into new plants. Okay. Simple. Vegetative method, it is done by fragmentation. So here what happens? The vegetative thallus, it breaks accidentally into new fragments, and these new fragments, they grow into new plants. Okay, you can see the older parts die and decay, separating the younger parts. So this is accidental, or sometimes when it happens, they later continue to grow and finally develop into full-fledged sargassum plant. So example, Sargassum natans, Sargassum hystrix, they multiply exclusively by this method. So the method of vegetative reproduction here, it is by fragmentation. Now next is sexual reproduction. Now here the sexual reproduction is of oogamous type. Why oogamous type? Because we have two sex organs, that is antheridia and oogonia. Now here, the sex organs are produced in flask-shaped cavities and they are called as conceptacles. I told you in the structure. Okay, the sex organs produced in flask-shaped cavities called conceptacles, and they are born on the sporophyte plant. Development of conceptacles. Then we have development of sex organs. For male, it is called antheridia, whereas in female, it is called as oogonium. Then after that, we go under fertilization, and then we have germination of zygote. This step by step we will study. Okay. Now, development of conceptacle. How from what they say, fertile branch of receptacle, a conceptacle is formed. So this is a development of conceptacle. I will tell you, explain you one like point wise. Just understand seeing the diagram, or you can also note it down. Okay, so here the conceptacle, it may contain either antheridia or oogonia. Okay, hence conceptacles are unisexual. So here you have to understand both the sex organs, male and female, are not found in one conceptacle. One conceptacle has only either antheridia or oogonia. Okay, the conceptacle containing antheridia, it is called male conceptacle, and conceptacle containing oogonia, it is called as female conceptacle. Clear? Male conceptacle is called as antheridia it is called as male, and oogonia it is called as female. The surface of the male conceptacle is smooth. Okay, but in spinous, whereas it is when the conceptacle is spinous, it is called as female conceptacle. Okay, now plants of sargassum are both monoecious and dioecious. Like only in one plant you can find both the sex organs, or you have two plants, one is having antheridia and another is having oogonia. Then next, in monoecious plants, the male and female conceptacles are produced in the same plant, so that is called as monoecious. And in dioecious plant, the male and female conceptacles are produced in separate plants, so that is why it is called as dioecious. Okay. Now, how development of conceptacle takes place? Now, each conceptacle, it is a cavity. Okay, it is a cavity embedded in the fertile receptacle. I told you from fertile receptacle, each conceptacle arises. Okay. The wall of conceptacle is 1 or 2 cells in thickness. You can see the diagram A here. Diagram A. The wall of the conceptacle is 1 or 2 cells in thickness. The wall cells are compactly arranged and they are rich in chromatophores. The wall cells are compactly arranged and are rich in chromatophores. The conceptacle opens to the outside by the means of ostiole. You can see diagram G. Okay, diagram G. The floor of conceptacle may be unbranched and extrude, or the conceptacle opens to the outside by the means of ostiole, and the filaments are called as paraphysis. So these basal cells are also called as paraphysis. The paraphysis is made up of colorless and barrel shaped cells. Okay, so this is about conceptacle. You just remember conceptacle may be either male or female. They cannot be both. Then the opening of conceptacle is ostiole. If the surface is smooth, then it is male. If it is spinous, then it is female. Okay, remember this much. This is much is enough. Next, these are all the points through which whatever I've explained right now, these are all its points. I'll just read it. If you want, then you can note it down. We'll go very slowly because you have to understand each and single point. Whichever is important, I'm telling you. Okay, so here the conceptacle develops from a single superficial cell on the receptacle branch. Okay, receptacle branch means fertile branch. This cell called conceptacle initial is flask shaped and it differs from the adjacent cells due to larger size and prominent nucleus. The initial cell divides slower than the other cell. As a result, it gets lower in position than adjacent cells. So how does the development takes place? It enlarges in size and it becomes different from the adjacent cell. Initial cell divides by transverse division. Two cells formed are separated by a curved septum. The lower cell is called basal cell and the upper cell is called as tongue cell. Okay, when it divides transversely, you have two cells, basal cell and tongue cell. Then, the tongue cell divides transversely. The tongue cell divides transversely to make small filament which later disintegrates. The basal cell undergoes many vertical divisions to make fertile layer of the conspectacles. The cells of fertile layer later form antheridia and oogonia. The fertile conceptacles, the cells of basal layer do not spread in the upper part. This forms a narrow opening called ostiole. Okay, don't get confused. It is point wise. When you read it once, you will understand it together. This explanation might seem difficult, but if you see the notes and read it, it will be very easy for you to understand about the basics of conceptacles. And this is asked for 2 marks. Okay. Now, through conceptacles, we have two organs, that is male and female. Now we will see female conceptacle and male conceptacle. Through male conceptacle, there is there arises antheridia, and through female conceptacle, there arises oogonium, that is also called as egg. Now we will see male conceptacle, that is how antheridia is developed through this male conspectacle. Okay. See the diagram. The antheridia are produced in male conceptacles. Okay. The basal portion of the paraphysis acts as stalk of the antheridium. Now, please concentrate on the diagram H. As I am explaining you, you just see the diagram. It becomes easy. The basal portion of the paraphysis acts as a stalk of the antheridium. Each branch of paraphysis bear 2 to 3 antheridia. The mature antheridium is small ovoid structure that contains 64 male gametes or spermatozoids. Okay. The protoplast of the antheridium, it first divides meiotically and then mitotically to produce 64 biflagellate male gametes. So what happens? First, this antheridial protoplast, it divides meiotically and then it divides mitotically. After this division, you have 64 biflagellate male gametes. Then what happens next? The mature antheridia, they get detached from the paraphysis and they come out of the conspectacle through ostiole. Okay. Next, the wall of the released antheridia becomes gelatinized so that the male gametes are released free in the water. So once the anthrazoid is matured or the antheridia is matured, they come out of the ostiole and anthrazoids are released in water. The male gamete is pear-shaped uninucleate and it has two unequal lateral flagella. You can see diagram G. The other flagella is long and directs upwards and one flagella is short and it directs upwards. The body of gamete contains haploid nucleus, eye spot, chromatophores and blepharoplast, which helps in the movement of spermatozoids. So this is about antheridia. Here you just need to understand diagram H. Paraphysis, conceptacle wall, which is forming stalk. Then you have mature antheridium. Then you have ostiole through which the anthrazoids come out. Now this anthrazoids has two flagella, that is biflagellate. One is pointing upwards and another is pointing downwards. Okay, it is easy. Just understand the diagram and write your points according to the diagram. You no need to study about the development of antheridia, but only diagram H is enough. Now we have seen male. Then let's see female. What happens? Now these oogonia are produced in female conspectacles and these bears 2 to 8 oogonia in them. Okay. The diploid nucleus of young oogonium, the nucleus, first it divides meiotically and then mitotically. Then it produces 8 haploid daughter nuclei. Okay. Then out of these 8, 7 degenerate and only 1 matures. So what happens? Understand. Now, there is a diploid nucleus in oogonia. Okay, so there is a nucleus in oogonium. First, it divides meiotically. Okay, and then it divides mitotically. So then total 8 haploid daughter nuclei are formed. So out of this 8, what happens? 7 degenerate. Matlab 7, they become functionless and only 1 becomes mature. And the one that becomes mature, it turns into ovum or egg. Okay. Now, what happens after that? At maturity, Okay, they uh, project out of the conspectacle. You can see diagram F. The egg is projecting out of the conspectacle. So this is the cavity of conspectacle, diagram F. But after maturity, what happens? It comes out. It comes out. It projects out of the conspectacle. Okay, and it remains attached by gelatinous stalk. You can see here the gelatinous stalk is there. Diagram F. Now this oogonial, it is made up of three layers. Three layers: outer layer, inner layer, and middle layer. The outer layer is called as exochiton. Then the middle layer is called as meso, and the inner layer is called as endo. Exo, meso, endo. This is easy to remember. Okay. Now remember, the egg is haploid, right? And the spermatozoid or anthrozoid, it is also haploid. Next, how fertilization takes place and how there is a development of a zygote. Now, what happens? Now you have haploid anthrozoid, and you have haploid egg. Now, how fertilization takes place? The released male gametes swim in water and they approach oogonia. So, the anthrozoid approaches the egg. The male gamete or spermatozoid, they are attached to the egg by their anterior short flagella. I told you spermatozoid has two flagella, that is biflagellate. The one which is short, it attaches to the egg. After attaching, what happens? The sperms, they penetrate into the oogonial wall and it fuses with the egg and forms a diploid zygote. Now, sperms are released in water. So, this sperm comes and attaches to the egg through the to the oogonial wall. It penetrates into the oogonial wall and it fuses with the egg. And after that, it forms a diploid zygote. Okay. So, this diploid zygote is called as a, like it is called diploid and it is called a zygote. Now you have zygote. What next? D, you have combination of male and female. Now how it develops? Okay. The zygote starts germination without rest because during favorable condition it develops. The zygote divides transversely into upper cell and lower cell. Diagram E. It divides transversely to form upper cell and a lower cell. The upper cell, by repeated division, it forms a vegetative part of the thallus. Upper cell, it forms the vegetative part, and lower cell, it forms rhizoid, then it forms hold fast. So that results in a diploid sporophytic plant. Understood? This is zygote. This divides transversely. It forms upper cell and lower cell. The upper cell forms the vegetative structure and the lower cell forms rhizoid and basal part, that is also called as hold fast. So then as a result, you have a diploid sporophytic plant. Okay. So by this we can say the life cycle of sargassum is diplontic life cycle. That is, the vegetative thallus is a diploid sporophyte. Okay. Now, just the conclusion, I'll just tell you one point wise, like from what we studied. The sargassum exhibits diploid, that is diplontic life cycle. Then the vegetative thallus, it is a diploid sporophyte. Then it produces male and female sex organs called antheridia and oogonia. They undergo meiosis and they produce haploid male gametes and haploid female gametes. The male and female gametes unite to form diploid zygote, which develops into diploid vegetative thallus. Here, the sporophytic phase is dominant in the life cycle. Now, the gametophytic phase, it is represented only by gametes, that is eggs and spermatozoids. So, the life cycle is called as diplontic life cycle. So, here there is no alteration of generation. So, in sargassum, there is no alteration of generation. Okay. Now, this is a graphical life cycle of sargassum. What is happening? Vegetative reproduction takes place by fragmentation, and sexual reproduction is by oogamous type. So in oogamous type of reproduction, what happens? You have male conspectacle and female conspectacle. From female conspectacle, you have oogonium, and male, you have antheridium. From oogonium, you have egg, and from antheridium, you have anthrozoid. They together fuse to that undergo fertilization and form zygote. And from zygote, you have a vegetative structure, that is sporophytic plant, which is diploid, and that gives rise to a sargassum plant body. So this is about the life cycle of sargassum. Here, the main important thing to remember in sargassum is conspectacles. Please remember, write it down somewhere. Conspectacles, chiroblast, chiroblasts. These are for 2 marks. Conspectacles of sargassum can be asked for 5 marks, wherein which you have to write 2 types, male and female. Then write how antheridia and oogonia are produced. It is very easy. Likewise, oogonium only see when it after antheridia and oogonia, everything is the same. But how antheridia and oogonia are produced, that is different. Okay. Here in sargassum, how they are produced? They are produced in a structure called conspectacle. That you have to remember. Everything is same. You can see this graphic cycle is same, only here this portion this portion below, like before meiosis, it is different in every species. After that, it is same. Like you have sperm, you have egg, they undergo fertilization, they form zygote, then germination of zygote takes place. This portion is same. Only thing is how they are produced, that is different. Okay. See, this is not at all hard. Please read the notes and you will understand it. Okay. Now tell me if you have any doubt. Yes, students. Okay. I hope you have understood sargassum. Take your time, read about it. Next class, we will study batrachospermum and economic importance of algae. So please read sargassum, because today only if you read, you will just at least remember the diagrams and get to know the points. Otherwise, you'll just forget it and you won't remember it. Please write the points. Please write 2 marks, 5 marks. Today, please study about conspectacles. Okay. That is important. Study today only, understand its point. If you have any doubt, ask me. next class. As and wise in WhatsApp groups, you can post it. Okay. I hope you all have a very good day.